concepts, and this has led to the development of different models. And these models, why we are doing that, I'll be telling you about that, why we are studying these models. I think this slide is not changing. Am I audible?
Yes, ma'am, audible.
But the slide is not changing. Yeah. So why do we need these different models? Why are we studying so many models? We are getting new scientific data, and we want to incorporate this in our current understanding of the disease and how the disease is initiated, how it modifies, and what are the different resolving factors. And these different models will give us an idea about how to take control of the disease. So the first model that was given was the linear model. This is what we were actually thinking, that let us have one bacteria and let it create the disease, let it have the disease, and we could take care of this. So this was the linear model which was initially given by Low and this in 60s. In 76, this is the first time that histological studies of our periodontitis was made, and Page and Schroeder in 1976 gave the classical model of differentiating this disease into 4 stages. We had the initial stage, we had the early stage, established stage, and advanced stage, and we had different types of cells coming in and creating more and more inflammatory exudate there, and getting the disease and creating the attachment loss. So this was the typical model that was given at that 70 time. And let me tell you, even after so many years of research, we still have this model, Page and Schroeder's model, as the basis of our understanding of periodontitis. So it actually had the first coming in the neutrophils and the T cells, B cells, and ultimately the loss of bone that led to the loss of the attachment, and that led to the periodontitis, development of periodontitis. So this model, I think all of us know. Then came the Sarka model. Now, there was the research on microbiology going on for a long time, and we were able to see which type of microorganisms were involved. And we, for the first time in the Sarka model in 80s, the role of the gram-negative bacteria was first established as an etiology of periodontal disease, that we said that it shifts from the gram-positive aerobes to the gram-negative anaerobes. This was the first time established in 80s. And we thought that this was a model which was going to explain everything. And we also had a study of protective and destructive nature of the immunoinflammatory components in the local tissues. So this circular model was developed. After that, in 1996, Offenbacher gave a model which not only established the etiology of the disease as a gram-negative one, it also thought about why in some patients there are variable sites involved, and why some sites are involved more than the other sites. So this model actually gave an insight into some critical parts of molecular and cellular biology or pathogenesis, which explained the disease in that. So there was a poor oral hygiene. It led to the pathogenic flora, and it later on went on to having the disease. So the most important thing in this model, in this model, was critical pathway model, and later on the composite model, was that there were some areas which were critical for the development of disease. For example, if there is an antibody neutrophil axis and this goes errant and erratic, then the disease will develop. If it's stopped at this place, it will not go to the further breakdown of the tissues, because we actually saw in some patients with the same amount of plaque and same amount of pathogenicity of the plaque, there was not much involvement of the periodontal tissue. So we, for the first time, thought that there is something contribution from the host side of the disease. And this also gave another component which was important from the advancement of the disease, that was the monocyte lymphocyte axis. So if these two axes were actually impaired, or they were deranged, then only the host got the disease. And for the first time in this model, in the composite model, we gave the idea of the contribution of other factors, that is the stress, genetic traits, diabetes, and smoking. This also contributed to this. Then in 1997, this was 96, 97 came the classical model. Now this could explain most of the processes that are occurring in the development of periodontitis. This gave the idea about that microbial challenge basically affects the host response, and it causes the connective tissue disease, and that is how it develops the this. And now in this, we thought about that there's more contribution from the host rather than from the bacteria. This was the classical model, and this was the first non-linear model, which is not that directly the bacteria is causing disease. So this was the model which gave an idea about the intricacies of the cytokines, prostanoids, matrix metalloproteinases, collagenases, and their interaction, and the disease, whether it can be stopped at some point, depended upon how much in the milieu the cytokine, pro-inflammatory cytokines were there, how many anti-inflammatory, what was the concentration of anti-inflammatory cytokines. So this gave an idea about the milieu that decided whether the disease will go into the progression or it will be stopped only at the gingivitis level. Again, some environmental, as in the previous composite model also, we had the introduction of environmental factors, stress, diabetes, and smoking. In this model also, the contribution from environmental and acquired risk factors was explained, and it also gave us an idea about how the clinical science developed over a period of time. Then came the ecological model. Now the people who are not researchers were not only looking at what is happening at the disease process, they were also studying the bacteria at a very metabolical level, and they would actually relate some mechanisms that some of the products from some of the bacteria of the primary colonizers were actually used as the growth factors by the secondary colonizers. And and they also found out that the most of the organisms that were involved for the breakdown of tissues were the proteolytic, the bacteria which had proteolytic enzymes. Those were the bacteria. For example, Porphyromonas, it uses heme as a heme as the growth factor, or Privetolite uses the sex hormones as the growth factor. So they also established the reason that not only the bacteria's presence is there, the importance is also that the bacteria should be able to attach to the surfaces, and that actually was considered as a virulence factor of that bacteria. If the bacteria is able to attach to the local tissues and deliver the toxins or the virulence factors from the local factor, that is the contribution from the local factors. So this also led to the, at the same time, there was a lot of research going upon on the omics, for example, genomics, metabolomics, or proteomics. So this also led to study of the local milieu at a more composite level. Composite level, I mean to say, they were not now studying only one bacteria and its virulence factors. They were looking at the whole of the milieu by taking out fluids, or by taking tissues, or by taking saliva or GCF, and studying what is the whole genome of that, what are the metabolites in the whole of that, not only from one organism but whole of the area which was actually having the breakdown of tissue. So there were advancements in that, and that led to the development of the biological systems model. So this system basically concentrated more on the overall sum total of the components and the virulence factors of bacteria, rather than only studying one bacteria and its effects. Now, this biological system in 2008, this model was given, which actually gave an idea about what is happening at the personal level, what is happening at the genetic level, what is happening at the biological phenotype, and all these together will ultimately decide what is the clinical phenotype. This is only possible if we are studying the milieu, and we are not studying only the organisms. If we are studying the milieu, that milieu, the local tissues, GCF, saliva, and other things, they will give an idea what is the contribution from smoking, diabetes, obesity, biofilm, or age, gender, and all the biomarkers. What is their contribution? What is the local concentration of each of these components, or the outcome of these personal and genetic levels? And that gave the idea of what phenotype will develop. So this was explained in a very intricate way, and they studied the intracellular component. They studied how the signal transduction was there, gene expression was there, what was the level of protein expression, and which cytokines were actually being manufactured in that local area, what was the tissue that was involved, and what were ultimately the clinical outcomes. So if we look at this diagram, so at different levels, it progresses from the subcellular, that is the microscopic, submicroscopic components, and it expresses in the clinical outcomes only as the different components affect the cellular system, or the cellular manufacturing of the cytokines or the metabolites from that local area. So if we look at this diagram, this is a very important diagram. If you look at this diagram, this is the clinical representation. And if we see the contribution from microbes, from the immunoinflammatory response, from the bone connective tissue, and from the host genetic variation. So each of these components, if you see this one plate, there is contribution from each of these areas, and then only this whole sum area will decide what clinical representation there is. So this gave an idea about what is the contribution, how much contribution is from each of these components, which is ultimately affecting the outcome or the clinical presentation of the disease. So this was how it was given in this model. Now, this also, this was the contribution from different areas. And then in 2012, HEATON and DIETRICH gave a model which is also known as the generic multi-causality model. Now, he tried to simplify the same thing what Offenbacher had given, that he was actually representing it, they represented it in a pie diagram where the contribution from the genetic factors, the lifestyle factors, environmental factors, systemic disease, and other unknown causes were represented in a pie diagram. So if we look at this pie diagram, we can easily find out that if we are looking at what was previously called as the aggressive periodontitis, we will have more component of the genetic component in that. So we can have a bigger circle or a bigger pie from the genetic component in the people who are having either the aggressive or what we call now very severely or rapidly progressing periodontitis. So in those patients, we could make a pie diagram according to the contribution from each of these. Now, at the same time, there were concepts. Now, in the first 2-3 slides, I gave you a concept that we actually wanted to have one etiological factor, and we wanted to have the vaccine or antibiotic against that one causative organism. We were always, so researchers were always trying to look for that one magic etiological factor and one magic bullet to control it. So in the same way, in the same research process, Sokransky and co-workers actually gave the idea about the different complexes, and they gave the contribution from different complexes. As we know, red complex actually represents that there is bleeding on probing, and these are the anaerobic bacteria, and they contribute a lot. They have a lot of virulence factors, which not only causes tissue destruction, which also evades the host response to phagocytose them. So they give this concept because at the base of it, we always want to have one etiological factor. So there was a continued interest upon whether there are pathobionts and there are keystone pathogens. Then we thought that P. gingivalis is the keystone pathogen, and it causes other bacteria to change into pathobionts, and they ultimately cause the local dysbiosis, and that is how disease is called. So this was the first step towards the concept of dysbiosis. So, even if we are thinking about this, that there are a group of organisms, a very small group of organisms which is causing the disease, then we should have been able to have an antibiotic against that. But in many studies, we have seen that even the treatment with antibiotic is not absolute. The disease returns, and we are not able to find that magic bullet. So this was an attempt to find one organism or a group of organisms which could control. So the concept behind that was that. So what did this say? The HAJISHENGALLIS actually gave two theories about the pathogenesis of periodontitis. It was one in 2012 and another one in 2014, and they actually explained how different types of organisms change. So what they basically were saying, that the body is usually having, these are all endogenous organisms, nothing is coming from outside. So body is living in a symbiotic relation with all the endogenous organism. But then there are some factors, maybe some environmental factors, local immunoregulatory defects or smoking, stress, that changes the local milieu, and this changes these commensal bacteria into pathobionts. There is dysbiosis, there is the immune system is not able to regulate that, even if in health they were the same organisms that were present there, but now the immune system is dysregulated, and the bacteria are using the immune system for their advantage rather than They are rather than getting the body rid of these organisms. So they also studied about the effect of gene expression and indirect effects on the community via host modulation. They also said that these organisms were able to change the way the host is reacting to the organism. So this was the first time that they were concentrating on the dysbiosis, this concept in 2012. So what they said that P. gingivalis is the male culprit. That's why we thought that we would actually manufacture a vaccine against the P. gingivalis, and that should actually treat the periodontitis. But we were very disappointed with the outcomes of the introduction of vaccines, because still it couldn't treat the disease, because that was just controlling one factor in that, which was maybe not even the primary factor for the disease, and there were many secondary factors and even other primary factors which we were not looking at. So they thought that how this P. gingivalis not only causes destruction by its virulence factors, it also causes TLR4 and deactivation, so that the body, the cells, the macrophages and the other antibody, these immune cells could not recognize the bacteria, and that if they don't recognize the bacteria, they cannot phagocytose them. And another part is that TLR2 is an important component on the macrophages and neutrophils, which gets attached to the bacteria, and it causes the formation of an array of cytokines, and it causes the formation of complement system, and that is how the disease is controlled. Now this organisms was also put as a culprit for actually subversing the crosstalk between the TLR2 and complement system, so that the body could not recognize it as non-self, and they would evade the immune system. So it actually triggered all these factors. They triggered the inflammation, but that did not lead to the resolution of the disease, and that only led to inflammation, which was very beneficial for the organisms, because then they had a lot of nutrients for their survival and for their growth and replication. So this concept also was there for a long time, and this still explains some of the pathological processes that are going on. And where did this come, this dysbiosis come from? How did we reach this, that we should extrapolate this, interpolate this on the periodontitis? This was the first term that was given in 1958, but that was not related with periodontitis. It was related with the gastrointestinal disturbances in the infants. And but we are not able to put the same type of application in our periodontitis, because most of the studies that have been done on the dysbiosis, they have methodological and analytical limitation. And we for a long time have been actually grappling with this definition, what is the most accepted definition of periodontitis, and what is the time that we should take the samples? Do we actually know exactly when the disease is in an active stage? Are we taking samples then? Are we studying dysbiosis then? So these studies, which actually were proving that dysbiosis is the ultimate model of periodontitis pathogenesis, they had many limitations, and that limitations actually affected the outcome that they extrapolated. So this concept actually was about that there is a local personalized pathology, that a particular person with its components from stress, environmental factors, smoking, diabetes, and genetic variations, they have their own pathology. That is, bacteria are same almost in all the people, but personalized pathology actually relates with the variation in other factors that are contribution to the pathogenesis of these diseases. So, and also that there is a limited time when you can take the sample. So we may not be sure whether we are taking that sample exactly at the time. Bleeding on probing is one of the objective signs, but there may not be exactly at the time when we are taking the samples. This is always a dynamic process. Then in 2012 came the polymicrobial synergy and dysbiosis model. Now we were not putting this P. gingivalis as a culprit, main culprit. So we thought, again, we are going from one organism to multiple organisms. When we are going again to one organism or a group of few organisms, now again we are going to a group of organisms or more organisms. So what we were, what they in 2012 suggested that, okay, that dysbiosis is not exactly explaining the periodontal pathogenesis, let us work at a model which gives an idea about a group of organisms which are not abundant, which are very less in number, but they are the drivers, and there are passenger bacteria. So what do you mean by driver and passenger bacteria? Drivers are the ones which are less in abundance, but they actually initiate the process of dysbiosis, and then there are many organisms which take advantage of these driver organisms. They get the nutrients, they thrive at the nutrients, they get the genetic material from these. There is intercellular signaling between these organisms. They actually transfer the genetic material and make the other organism more virulent towards that virulent towards other organisms, more virulent, and that is how the disease progresses. So if we look at this diagram, so there was a cooperation between the symbiotic bacteria, and there was homeostasis, and there is immunity. There is health. That is that. Now, different factors, that is subversion of this organism causes subversion of host response. As I was telling you, this was the main culprit that was thought to be there. And then smoking, aging, diet, all these may lead to the actions where there is dysbiotic microbiota is formed. There's a positive feedback. This inflammation causes more bacteria to get the available nutrients, and then they will divide at the cost of the local milieu, and that is how the tissue breakdown occurs, and there is development of periodontitis. So this model. So what we have as the key factors in this model is that there are some keystone pathogens, which are the bacterial drivers. They cause dysbiosis. They affect the immune surveillance. They do not let the body see that. So what if we look at this from this aspect, if we look at, so there are, that now was the concept that there were some organisms which were not previously seen. They were Filifactor, Alosis, Selenomonas, Disulfugus, and these are the ones which were not cultivable. But since we have PCR and we can see the genetic composition of things, we can actually delineate some more organisms which may not be cultivable, but we can identify these organisms, and we can study their pathological processes. So what these pathobionts actually do, they exploit the nutritional supply to the local tissues, and there is a and ultimately they outnumber the keystone pathogens. That means the ones which initiated the disease, they were less, then they actually incited other organisms, and they became more than the primary organisms, and there were some accessory pathogens, for example streptococcus faundi, and they may have some synergistic relation with each other. If they are trying to have a cooperative relationship, then we call that black queen hypothesis, and if they have antagonistic interactions, then we call that red queen effect. So all this was basically directed towards a group of organisms which were the keystone pathogens. Then they incited the pathobionts, then they had interaction with other communities. Now this concept of gram negative and gram positive, aerobic, anaerobic, did not apply here, because here we were seeing that there were very few organisms which were following that primary colonization, secondary colonization, that type of effect. So this model actually called about the contribution from all the types of organisms, gram positive, negative, and their association. So this collective synergy between these organisms, that is the drivers and the passengers, is called as the nososymbiosity. This is a lived term which says that there is synergy and antagonism going on at the same time. So the notion that we were looking at, that one single or a few select organisms, that was dismissed in this theory, and we again were saying that there are multiple organisms. Now, because we are putting in a lot of implants, we have to think about how to define the pathogenesis in the periimplantitis. So here, apart from that, there is a homeostasis breakdown in the local milieu in that portion. We are also studying, because now we are dealing with something which is mechanical. We are also dealing about the material related factors have been incorporated in that. That is the roughness, wettability, and chemical composition. They also play a role in the pathogenesis of disease. So periimplantitis not only has this dysbiotic or this multi synergy or noso synergy symbiosis, they also have the contribution from the type of implant that we are placing. So the synergy and dysbiosis in periimplantitis also involves the material factors. So and now we in 2020 we had this model of revised model, that is we initially thought that there were primary colonizers, then there were secondary colonizers, and then the disease developed. But this revised model by Bartold actually talked about the talked about that the inflammation was there, there was a non-specific inflammation, and that altered the environment, and then we had the organisms that gave primary importance to inflammation and the acquirement of nutrients, and then the development of organisms or development of different types of colonies over there. Then came the another model, that is immunomicrobiological pathogenesis model, that is by HAJISHENGALLIS only in 2014. It is the same model where they explain about the dysbiosis and all that. But now, at this time, there was a lot of research going from the immunological point of view. Now you should see that there is the development of, we had initially what we had was Th1 and Th2 responses. They would explain whether the disease would progress or it would not progress, but they did not properly explain that some cytokines are leading to the breakdown and some cytokines are leading to the resolution of the disease. At this time, there was a big discovery, that was the development of this Th17 concept. There was Th1, Th2, but Th17 concept developed at that time. And in that light, they described the role of this new regulatory cytokine IL-17 and its role in the disruption. Now, this IL-17, you cannot typically place in either the pro-inflammatory or anti-inflammatory. It has some positive responses. It helps in the erythropoiesis. It has some advantages in the epithelialization of the tissues, but at the same time, it's basically a pro-inflammatory cytokine. So the regulation from Th17 cells was found to be one of the most important components of the disease progression. And this model of immunomicrobial pathogenesis model actually explained the role of this different type of new cytokine that was there. So if you look at the functions of cytokines, particularly this IL-17 cytokine that comes from Th17, then you would see that it not only affects the formation of these we always thought that neutrophils came first, and then they were actually they went out of. Hello, am I audible?
Yes, madam.
Actually, my
Yes,
computer
ma'am.
is saying that it's unstable. Is it okay?
Yes, madam.
No. Okay. In the patient Schroeder model, we always thought that neutrophils are the ones which are the first line of defense, and then over a period of time they go out of the sight. But with the new knowledge about the immunopathology of the diseases, we knew that neutrophils are not going anywhere. They are actually affecting the other cells there, and they are always there. So the new role of the neutrophils also came into the light, and it also explained how IL-17 is able to indirectly subverse, if you can see it inhibits the neutrophils, and it causes more destruction of the local tissues. Then came in 2015 came a contemporary model, that is given by MEYLE and CHAPPLE. What they said that again they said that the dysbiosis is there, and then there is frank dysbiosis. There is incipient dysbiosis and there is frank dysbiosis. The difference was that they gave the concept of that the inflammation, if inflammation is rich in nutrients like heme and peptides, they will initiate the incipient dysbiosis, but it will not go into the frank dysbiosis if the host is not susceptible. It's only in the susceptible host that the incipient dysbiosis, which initiated with what we talked about keystone pathogens, and we talked about pathobionts, and we talked about the conversion of gram positive to gram negative. This would not go there if the host was not susceptible. So this all the more laid more emphasis upon the local milieu and the type of response that the host is developing towards a particular type of a organism. So this had many components in clinical health. There was if there was complement formation, PNMs, and they could effectively control the disease. In gingivitis, there was some type of dysbiosis, but it was not a frank dysbiosis. It was an incipient dysbiosis, and over a period of time, maybe with the control of inflammation and resolution of the inflammation, this could be controlled. But in periodontitis, once it developed to a frank dysbiosis, because of the risk factors, as we see in this also, the risk factors are increasing. That dysbiosis, which is now frankly there, which is giving rise to the pathogen biofilm, that has more of inflammatory responses, and that is how there is it's leading to the destruction of the tissues. So this gave an idea. So this model also gave us an idea about this. If you look at this diagram, that if the host bacteria, these are in the balance. If they go aberrant, there is some zone where their body can still take. There is aberration, that is what I was telling you, incipient dysbiosis. So there is a zone where the body can still handle. If there is a bit of dysbiosis, the body can handle that dysbiosis and it can effectively cause the resolution of the disease. But if these factors, host defense goes low and bacterial pathogenesis goes more, then even the accommodation by the host is not there to control the disease, and then it goes into the frank progression. So it goes from here to the progression of disease rapidly. So here up to here, there is a resolution. Even if there is some amount of organisms there, there will be resolution. There is some accommodation door, and then when it goes out of control, then only the disease progresses at a very rapid rates. So this actually gave the concept of proportionate and pre-resolving to non-proportionate or disproportionate to non-resolving. So this is going from this health towards the disease when it's progressing at a very rapid rate. So this was a concept given by that. So in 2015, again another model came. This is also same as the initial model of polymicrobial model, but also it gave the idea about the genetic component of that. This is what I was telling you, that there is the transfer of some genetic material from one bacteria to another, which makes one virulent species make other also species which were otherwise benign, they make them also very virulent. So this concept gave the idea about how genetic transfer between the different cells, between the dysbiotic microorganisms actually contributes to the genetic pole over there, and which is very important from the pathogenesis of the periodontal disease. So this also gave it that. Now there is in 2017 again HAJISHENGALLIS gave the concept of revised Page and Schroeder model. Now we were this is what I wanted to emphasize on particularly, because in Page and Schroeder they also thought about the role of neutrophils in the initial stages, and they thought that after the neutrophils, like we usually say, the neutrophils can phagocytize only 10 pathogens, and after that, if there are more, macrophages have a higher capacity of phagocytosing the organisms, and they can kill 20 or more than that. So that concept is there, and the neutrophils actually are apoptosed and they go out of the site, but nothing like that actually happens. Looking more keenly and more observantly with new techniques, confocal microscopy and all that, we are able to see that neutrophils don't go anywhere. They are there and they have a crucial role, not really only in the progression of the disease, but even up to the level of the bone loss, because they regulate neutrophils, they regulate lymphocytes, they are important in the rankle mediated bone destruction also. So now this model was actually revised, because it is the primary. I was telling you a Page and Spreader model is the primary model which explains the mode of disease, but with more advancement we are able to have a more insight into how this model explains the disease. So there's a defective surveillance because of these organisms, and it is shifted to the disruptive homeostasis. So now we know that neutrophils not only have a role in just phagocyting the organisms, they have a role in the on the natural killer cells. They have a role even in the T cells. They can go back to the lymph nodes, and from there they can actually select. They have an effect on the selection of the types of T cells that should be related there, and they even have a role in the stimulation of IL-7 Th17 cells, which can later on produce IL-17. So they have a role not only there. So this is a very very complex diagram, whereby actually I wanted to tell you about that the neutrophils have a role. They have a role over here. They even have a role in the osteoclastogenesis. They have a role in the maturation of the B cells or in the T cells. They have the role in the stimulation of these and the elaboration. So they are involved in even antibody production and rankle dependent and independent osteoclastic activity. So there is nothing like a very sharp line between the innate and adaptive immune responses. They go hand in hand and they are in a continuation rather than one role taking over by that. Now, explaining all these models, we have an idea about what is dysbiosis. I was telling you dysbiosis actually doesn't explain any much about how the disease progresses. That we are saying that there is a shift in the inflammation, and that is how there is altered growth environment. That is how there is different types of organisms they take over. If we think about the dysbiosis from the traditional concept, and we think about periodontitis, they do not actually explain the situation by the dysbiosis. And if we see the longitudinal integrated multi-omic studies, they are needed. More of these studies are needed to clarify this. Now, this is from a historical perspective, from an evolutionary perspective. For example, if we know that oral microorganisms, when the child is born, he has a sterile mouth, and slowly over a period of time, the organisms develop. Which are these organisms? They are the same organisms that are endogenous. They don't come from outside. So how did suddenly we thought that they are pathogenic and they are not living in a symbiotic relationship? If we study the historical perspective of our periodontal disease, the lifespan was not this much, and this was considered that the diversity in the microorganisms developed, but till they reached the bone loss, the person was no more there. So we couldn't study what is actually happening, because the lifespan of the people have increased over a period of time. That's why we are now looking at the pathogenesis of periodontitis from a different perspective. We are looking at it from the pathogenic point of view. Otherwise, in the histological perspective, we are actually looking at the organisms which are actually endogenous to the person. So if we study the other types of diseases, for example, if we are studying cholera, tuberculosis, or other types of phthisis and all that, what we are seeing that there was more diversity, and then it led to the low diversity. There was a high diversity of organisms, and then it led to a low diversity. Here, what we are seeing, we are here talking about more diversity. So how does it explain disease? If we are thinking that the harmless or the beneficial bacteria are less and the virulent are more, and we are getting disease. But if there is more diversity, that should not produce disease. It should be one organism actually multiplying at the cost of other organisms, which we don't see in periodontitis. In periodontitis, we see as the disease progresses, it goes to more diversity. So how does it explain disease? This disease can be explained in the low diversity only in the light of the aggressive periodontitis, where we do see AA in a lot of that. So if we look at this aspect of that, it doesn't explain. We have many drawbacks to that. We have seen that it is a polymicrobial insult, and we have many organisms which may be unidentified and have never been cultivated. The number and identities of the member bacterial species of dysbiotic periodontal microbiome differ between individuals. We don't have the same type of dysbiotic in all the individuals, and they do not, even if there is diversity, there is dysbiosis in that, it doesn't cause disease in all the organisms. So there are many drawbacks to this dysbiosis type of an organism. Look at this diagram. So if we have streptococci, crods and all that, can you see that there was less diversity when it was in health, and there's more diversity when there is in disease? This doesn't explain the disease on the basis of how we are explaining disease in the rest of the body. So more diversity is in the chronic periodontitis. So This, in the historical perspective, was a normal age-related mechanism which caused some loss of the tissues, and it did not lead to the alveolar bone loss because the person did not survive up to that time. It only explains diseases in the aggressive periodontitis, while we do see that there is less diversity organism, and there is the organism which is ultimately there is of a more virulent capacity. That's this is saying that I am all powerful and I can destroy all the bone and the connective tissue over there. Now in 2019, we actually went 180 degrees. We thought about, now we had an initial idea in the revised model of Bartold that we said that inflammation is important for that. But in this model, in 2019 model by Bartold and Van Dyke, they said that inflammation is the only thing that is important. If there is some inflammation and there is the milieu is such that it's full of nutrients, there is the development of disease, because these organisms, emergence of periodontal pathogens will be there, and these organisms who basically are of a type which are proteolytic and asaccharolytic. That means they degrade the proteins. They are not carbohydrate loving, or they don't use carbohydrates as a nutrient source. So what this embodied model said, that there is plaque accumulation. We are again going to the non-specific plaque hypothesis. We are not talking about one particular organism. We are saying that a group of organisms will cause disease, they will not cause disease. There is inflammation, there is an epithelial breakdown, there is clavicular fluid flow, and it causes a local inflammatory area in that. There is dysregulated immunoinflammatory response to the organisms which use these nutrients, and that is how the disease occurs. And if this immunoregulatory stage is normal in a normal patient without any risk factors, then it will go only up to gingivitis. It will never progress to the periodontitis. So this concept gave us an idea that the primary thing is inflammation, and only the organisms, they are secondary to the primary development of the milieu over there. Then in 2020 also came the impede model. So this model actually was there because now we have changed the classification. So we needed a new model to integrate that into that classification. And this also was under the main idea about how inflammation is the primary driver of plaque-associated periodontitis. So this model basically says that plaque in health may go up to gingivitis, but if there is inflammation, there is genetic environment is diet or smoking or other things, this causes a stage 2 or polymicrobial type of a dysbiosis, dysregulated post-immune response. There is overgrowth of, that is secondary to the inflammation. There is overgrowth of periodontal pathogens. There is inflammation-mediated dysbiosis. So we are explaining these stages of the periodontal disruption or destruction according to the stages of the new classification. So late periodontitis, actually now all the factors are involved. Post-immune is dysregulated, inflammatory reactions are there, there is genetic involvement, there is environmental is unable to contain the infection, and it leads to the dysbiosis. So now we are explaining it according to the light of the new classification. So this is the same thing, that initiation is there is inflammation, late stage, that is the stage 4. It's also just. But why do we want to do this? Why are we going into the sub-molecular or subcellular level to study the disease? So this was an article in 2020 by HAJISHENGALLIS only, who explained that we need to know exactly which pathological mechanisms are going wrong and where we can contain the disease. So these are the different, this is a new concept about the post-modulation. At what, if we know these mechanisms properly, then only we can develop the agents or the components which can stop this disease at different levels. So this, at the breakdown, we can have doxycycline as a regulation for MMPs. We can have for the recruitment of inflammatory cells, we have different types of mechanisms that how we can control. We have anti-cytokine therapy, we can have probiotics in light of this new type of disease progression that we are studying. That's why we want it. So if we want to summarize this, I hope I did not confuse you more than what you initially started with. So if we summarize this, I would like to say four things about this. That initially we had this linear pattern. In 70s, we knew about the anaerobic bacteria and the hostess model. 1990s gave us an insight more into the immune system. And at this point, we were saying that 80% of the disease is because of the disruptive host response rather than only there is 20% contribution from the organism. So this was the concept in 1990. And later on, recently models, they took different omics and they developed proteomics and genomics, and they study the local milieu rather than going into the host response and the bacteria. They were studying all these local areas what were there, and the biological models actually integrated all these systems together for better understanding. So this is a diagram which explains the different 16 models that have been given. And I want to conclude by saying that understanding of the biofilm has really drastically changed over the past century. We need to look at the history also, but we need to look at how we have developed different techniques of studying the bacteria. And not only do we study the cultivable bacteria now, we also study the bacteria that are not cultivated. So we need a continuous research. We need to explore new pathways so that we can actually stop these mechanisms and have the resolution of the inflammation by different agents. And this should be a tailor-made preventive and therapeutic strategies for the patients rather than giving the same type of treatment to all the patients. Thank you. Thank you for the patient listening. Hello?
Yes, madam, thank you, madam. I think, madam, you very well from early models to newer models. So I am sure your lecture benefited for understanding the periodontal disease. Continuous research, so in future time we can be for the treatment of periodontal disease. Thank you, madam.
Thank you. Thank you for. Thank you so much.
Over to Dhaval.
Thank you, ma'am. I would like to thank our speaker for today's session, Dr. Nitya Pandit, ma'am, for this highly informative session, and also our moderator for today's session, Dr. Surekha Rathod, madam. Thank you, ma'am. At last, I would like to thank all the participants for joining this session. So once again, I thank you all. So
Thank you.
ending the session, ma'am, now. Thank you.
Thank you, madam.
Thank you, ma'am. Thank you so much. Can I leave now?
Thank you, madam.
Can I leave?
Yes, madam.
Oh, thank you.